Hidden within the complex chemistry of our nervous system lies serotonin, a molecule that quietly orchestrates our fundamental sense of well-being. It is far more than a simple switch for happiness. Scientifically known as 5-hydroxytryptamine, or 5-HT, this compound is actually an ancient chemical messenger found across the entire kingdom of life. As we settle in to unravel its secrets, feel free to support our journey into these relaxing facts by clicking the subscribe button. It is fascinating to realize that serotonin predates humanity by eons, appearing in the biological records of single-celled organisms billions of years ago. It is not unique to complex brains, as it thrives in the roots of plants, the venom of wasps, and the tissues of fungi. Even the stinging nettle uses serotonin as a chemical weapon, delivering a mild shock to anyone brushing against its leaves to deter grazing animals. In the plant kingdom, walnuts and pineapples contain high concentrations of this molecule, though eating them does not directly alter human mood because the chemical cannot cross the protective barrier of the brain. While it is famously celebrated as the molecule of mood, labeling it solely as a happiness chemical is a significant oversimplification. It acts more accurately as a stabilizer, smoothing out the peaks and valleys of emotional experience to create a baseline of contentment. However, the brain is actually the minority shareholder of your body's serotonin reserves. A staggering 95% of this chemical resides not in the skull, but within the lining of the gastrointestinal tract. Here, specialized cells known as enterochromaffin cells produce and store serotonin to manage the rhythmic muscular contractions of digestion. This massive localized supply highlights the profound connection between the gut and the mind, often described by scientists as the body's second brain. The influence of this molecule extends even further, acting as a critical component in the body's physical repair system. Platelets in the blood do not manufacture their own serotonin, but they scavenge it from the plasma to store for emergencies. When a tissue is damaged, platelets release this borrowed serotonin to constrict blood vessels and accelerate clotting. Ultimately, this multi-purpose molecule serves as the bridge to the night, functioning as the direct chemical precursor to melatonin. Without the serotonin synthesized during the day, the body cannot construct the hormone required for deep restorative sleep. This crucial relationship between our biology and the rhythm of the night remained a mystery for centuries, hidden within the complex chemistry of human tissue. The journey to identify this multi-purpose molecule officially began in 1935 within the quiet walls of a laboratory in Rome. A young Italian pharmacologist named Vittorio Erspamer was conducting a detailed analysis of the stomach lining, specifically examining extracts taken from the intestinal tissue of rabbits. He observed a curious, potent substance that caused smooth muscle cells to contract rapidly, a reaction that suggested the presence of a previously unknown biological agent. Because he discovered this compound exclusively within the digestive tract, specifically within the enterochromaffin cells, he gave it the name enteramine. He derived this title from enteron, the Greek word for intestine, believing he had found a chemical specific to the gut's internal movements. While Erspamer was continuously mapping the chemistry of the digestive system, a separate team of researchers across the Atlantic was unknowingly hunting for the exact same substance, though for different reasons. In the late 1940s, at the Cleveland Clinic in Ohio, a group led by Maurice Rappaport, Arda Green, and Irvine Page was investigating the underlying causes of arterial hypertension. They were desperately searching for a mysterious factor in the blood that caused vessels to constrict, thereby raising blood pressure. To find it, they undertook a massive logistical challenge, processing nearly 2 tons of beef blood to isolate just a few tiny milligrams of the active compound. When they finally succeeded in purifying the crystalline substance in 1948, they needed a name that accurately reflected its origins and its specific action on the vascular system. They looked to the clear fluid in which it was found, the serum, and the tightening effect it had on the blood vessel walls, which was to increase their muscle tone or tension. By combining these two descriptive concepts, serum and tonic, they coined the elegant name serotonin. At that moment in history, the scientific community believed they had simply found a powerful vasoconstrictor, a mechanical tool the body used to manage blood flow and clotting. It was not yet viewed as a mood regulator or a key to sleep, but rather as a hydraulic valve controller for the circulatory system. The connection between the gut and the blood remained unmade until 1952, when further rigorous chemical analysis revealed that Erspamer's enteramine and the Cleveland team's serotonin were in fact the exact same molecule. This realization was a pivotal moment in the history of biochemistry. It meant that the same chemical responsible for contracting the intestines was also circulating in the bloodstream to manage vascular tension. The molecule was identified chemically as 5-hydroxytryptamine, a name often shortened in medical literature to 5-HT. However, the most profound shift in understanding was yet to come. Shortly after the unification of these discoveries, a researcher named Betty Tworog demonstrated the presence of serotonin in the mammalian brain. This finding shattered the prevailing view that the substance was merely a peripheral hormone for digestion and blood pressure. Finding it within the central nervous system suggested a far more complex role, indicating that this simple serum tonic was not just moving blood or food, but was likely involved in the sophisticated transmission of nerve signals, effectively acting as a bridge between the body's physical state and the mind's operation. To understand how this bridge is built, we must look at the blueprint itself, zooming in past the tissues and cells until we see the specific arrangement of atoms that gives serotonin its unique power. In the precise and rigid language of chemistry, this molecule is officially known as 5-hydroxytryptamine, a designation often shortened to just 5-HT. While the full name might sound intricate, it is actually a literal description of the molecule's architecture, effectively acting as a map that tells a chemist exactly where every atom sits in space. At the very core of serotonin lies a distinct shape known as an indole ring. You might visualize this as a flat, geometric structure where a 6-sided hexagon is fused seamlessly to a 5-sided pentagon, creating a double ring formation that serves as the sturdy skeleton of the molecule. This specific shape is deeply familiar to biology because it is almost identical to tryptophan, the essential amino acid found in your diet. Essentially, your body takes the raw material of tryptophan and performs a delicate renovation, attaching a hydroxyl group, a pair of oxygen and hydrogen atoms, to the 5th position of that ring. This subtle addition is the magic trick, the chemical shift that transforms a simple building block of protein into a communicative master key, capable of unlocking complex neurological responses. This structure places serotonin in a prestigious chemical family known as monoamines. The amine part of the name refers to a specific cluster of nitrogen and hydrogen atoms tailing off the main rings. This tail acts like a chemical handle, allowing the molecule to dock securely with receptors with satisfying precision. Being a monoamine means it shares a lineage with other heavy hitters like dopamine and norepinephrine, yet serotonin is unique. It is simpler than many pharmaceutical drugs, but far more specific in its action. Despite its microscopic size, the molecule is chemically tough. It is stable enough to endure the acidic and enzymatic chaos of the digestive tract, which is fortunate since that is where most of it is born. If you were to hold a model of it, it would look compact and balanced, a tight knot of carbon, hydrogen, nitrogen, and oxygen.
It is not a sprawling, fragile chain, but a dense, efficient tool designed by evolution to be manufactured quickly and to survive the journey to wherever it is needed, remaining intact until it slots perfectly into its destination. To create this dense molecular tool, the body employs a synthesis pathway that acts like a highly specialized biological assembly line, characterized not by haste, but by a precise rhythmic cadence. This manufacturing process begins with raw materials that the body cannot produce on its own. The genesis of every single molecule of serotonin in your system is an amino acid called tryptophan. Tryptophan is the essential starter block, a substance you acquire through your diet, found in seeds, nuts, cheeses, and meats. However, simply consuming this raw material is not enough to guarantee the production of the neurotransmitter. Tryptophan must first compete for transport. Because it is a large neutral amino acid, it shares a transport system with other amino acids like tyrosine and phenylalanine, effectively waiting in line for a ticket to cross the blood-brain barrier. Once the tryptophan successfully navigates this competitive entry and arrives inside the neuron, the true alchemy begins. The first step of this transformation is the most critical. It involves an enzyme known as tryptophan hydroxylase. In the world of biochemistry, this enzyme is the gatekeeper. It performs what scientists call the rate-limiting step. This means that the speed of serotonin production is entirely dictated by how fast this specific enzyme works. It acts as a natural throttle, ensuring that serotonin is produced at a steady, manageable pace rather than a chaotic flood. Tryptophan hydroxylase works by taking a molecule of oxygen and a molecule of iron, along with a cofactor called tetrahydrobiopterin, to attach a hydroxyl group, a pair of oxygen and hydrogen atoms, to the tryptophan ring. This subtle addition is the magic trick, the chemical shift that transforms a simple building block of protein into a communicative master key capable of unlocking complex neurological responses.
Their primary biological imperative was to find nutrients and process them. The nervous system originally evolved to coordinate this digestion. In a way, the brain in our head evolved much later, essentially as a navigational tool to help the gut find more food. We are, in an evolutionary sense, elaborate vehicles built to serve the needs of the digestive tract, and the dominance of serotonin in the gut is a chemical relic of that ancient priority. But this ancient chemical dominance is not a solo act performed by the human body alone. In fact, if we look closer at the lining of the intestines, we find that the production of serotonin is actually a collaborative effort, a partnership between our own cells and the trillions of microscopic residents that call our digestive tract home. This is the domain of the microbiome, a vast and complex ecosystem of bacteria, viruses, and fungi that outnumber our human cells and possess a genetic diversity that dwarfs our own genome. For a long time, science viewed these bacteria merely as passengers or potential pathogens, but recent discoveries have shifted that view entirely, revealing that these indigenous bacteria are essential conductors of the serotonin symphony. When researchers examine germ-free environments, specifically animals raised completely without gut bacteria, they find something startling. These creatures possess significantly lower levels of serotonin in their blood, sometimes 60% less than those with a healthy microbiome. This deficit suggests that without our microbial partners, our body's serotonin factories simply do not run at full capacity. The connection is specific and chemical, reliant on a sophisticated form of cross-species communication. Certain spore-forming bacteria, particularly from the genus Clostridia, act as direct supervisors to the enterochromaffin cells we mentioned earlier. They release specific metabolites, chemical byproducts of their own digestion, which drift over to the gut lining and signal our cells to ramp up production.
It is a strange and fascinating realization that the chemical regulating your mood and sleep might be dictated by the microscopic life living within you. And I wonder if you have ever intuitively felt this connection yourself, perhaps noticing how a specific meal seemed to alter your emotional state or mental clarity in a way that felt deeper than just simple digestion. These bacterial signals often take the form of short-chain fatty acids, fueling the very cells that synthesize the neurotransmitter. It is a symbiotic loop where the bacteria help regulate the host's motility, literally speeding up the transit of food through the gut, which in turn helps the bacteria spread and thrive. But the influence of these microbes extends beyond mere digestion, bringing us to the emerging field of psychobiotics. This revolutionary concept suggests that because gut bacteria modulate serotonin, they consequently modulate the mind. The term psychobiotic was coined to describe live organisms that, when ingested in adequate amounts, produce a health benefit in patients suffering from psychiatric illness. It sounds like science fiction, yet the biological pathway is sound. While the serotonin produced in the gut cannot cross the blood-brain barrier to directly enter the brain, it influences the brain through indirect channels. The primary channel is the vagus nerve, the long, wandering nerve that connects the brainstem to the abdomen. Gut serotonin stimulates the nerve endings in the digestive tract, sending electrical signals rushing up the vagus nerve to the brain, influencing regions responsible for emotion and stress response. In this way, the bacteria are essentially speaking to the brain via a hardwired telephone line, using serotonin as the voice on the other end. Research has focused on specific strains like Lactobacillus and Bifidobacterium, finding that introducing these bacteria can reduce cortisol levels and alleviate symptoms of anxiety, effectively mimicking the action of antidepressant medications, but through a purely biological route. Furthermore, these bacteria influence how we process tryptophan, the amino acid precursor to serotonin. Without the right balance of gut flora, tryptophan can be diverted down a different chemical pathway, one that leads to inflammation rather than happiness. This means the microbiome acts as a gatekeeper, deciding whether the raw materials we eat become the molecules of peace or the molecules of stress. But beyond determining our emotional state, this chemical gatekeeping dictates the physical rhythm of our survival. While the microbiome influences the production of raw materials, serotonin itself takes charge of the manual labor required to keep the digestive system running. In the enteric nervous system, this molecule acts as the primary conductor for the rhythmic, wave-like muscle contractions known as peristalsis. When food enters the small intestine, the physical pressure stimulates the enterochromaffin cells to release serotonin, which activates the nerves embedded deep within the gut wall. It signals the smooth muscles to contract forcefully behind the food and relax ahead of it, creating a traveling wave that propels contents forward. It is a precise, coordinated motion, ensuring that digestion occurs at exact the speed necessary for nutrients to be absorbed into the body.
If serotonin levels are too low, this transit slows down significantly, leading to sluggishness and constipation. Conversely, if levels are chronically high, the system rushes, resulting in urgency. However, the most dramatic display of serotonin's power occurs when the body detects a genuine threat. When the digestive system encounters something toxic, spoiled, or irritating, the gut's lining immediately identifies the danger. In a rapid bid to protect the rest of the organism, the enterochromaffin cells release an overwhelming flood of serotonin, far exceeding the subtle amounts used for normal digestion. This chemical surge serves two immediate defensive purposes. First, locally, it overstimulates the gut nerves to cause violent, rapid contractions designed to expel the offensive matter as quickly as possible, effectively flushing the system. Second, this excess serotonin floods the receptors on the vagus nerve, the massive information superhighway connecting the abdomen to the brain.
The vagus nerve carries this urgent distress signal all the way up to a specific region in the brainstem known as the area postrema, or the vomiting center. Because this part of the brain is uniquely sensitive to chemical alarms, it immediately initiates the sensation of nausea and the reflex of vomiting. While we often experience nausea as a miserable symptom of sickness, it is actually a sign of a highly intelligent, functioning defense system. It is serotonin's way of ensuring that harmful toxins are ejected before they can be absorbed into the bloodstream. In this light, that wave of sickness is not a failure of the body, but the work of a molecular guardian aggressively protecting its territory. But this protective vigilance is not confined to the digestive tract. If we follow the serotonin out of the gut and into the circulatory system, we discover another layer of its physiological brilliance hidden within the microscopic components of our blood. Here, the chemical acts as a critical structural engineer, maintaining the integrity of the body's plumbing. This role is primarily executed by platelets, the tiny, colorless cell fragments responsible for clotting. There's a fascinating dependency at play here. Platelets are unable to synthesize serotonin themselves. They lack the necessary enzymes to build the molecule from scratch. Instead, they act as mobile storage units. As blood passes through the gut, platelets scavenge the serotonin released by the intestinal cells, using specialized transporter proteins to vacuum it up from the plasma. They pack the chemical away into dense, secure compartments called granules, carrying it through the veins like cargo ships transporting ammunition, waiting for a specific signal to deploy. That signal arrives the moment a blood vessel is damaged. When you suffer a cut or a scrape, the smooth interior lining of the vessel is breached, exposing the collagen underneath. Platelets immediately sense this disruption and rush to the site, changing shape from smooth discs to spiky, sticky spheres that latch on to the wound. As they clump together to form a mechanical plug, they simultaneously unlock their granules, dumping their stored serotonin directly onto the surrounding tissue. The effect is immediate and mechanical. The release of serotonin causes the smooth muscles within the blood vessel walls to contract violently. This process, known as vasoconstriction, effectively narrows the diameter of the vessel at the point of injury. It is the body's way of applying a tourniquet from the inside. By tightening the pipe, serotonin restricts the flow of blood to the damaged area, drastically reducing blood loss, while the slower, more complex clotting factors work to weave a permanent mesh over the tear. Without this rapid, chemically induced constriction, even minor injuries could result in dangerous levels of bleeding. Furthermore, the serotonin molecules serve as a chemical beacon, signaling nearby cells to begin the process of tissue repair, stimulating them to divide and rebuild the broken wall. It is a stark reminder that before serotonin was ever involved in complex human moods, it was, and remains, a fundamental tool for physical survival, stitching us back together whenever we break. Yet this restorative power comes with a fascinating caveat when it turns to the solid framework of the skeleton. For decades, the interaction between serotonin and bone density remained a mystery, but we now understand that the gut and the brain have opposing views on how strong your bones should be. While the tiny amount of serotonin produced within the brain acts as a signal to build and strengthen the skeletal structure, the serotonin circulating in your blood, which makes up the vast majority of the body's supply, does the exact opposite. It binds to specific receptors on osteoblasts, the industrious cells responsible for laying down new bone tissue, and essentially tells them to stop working. This creates a necessary biological tension. If the gut produces too much serotonin, it can inhibit these building cells too aggressively, leading to a decrease in bone mass. It suggests that the body uses this molecule to prevent the skeleton from becoming excessively heavy or dense, maintaining a lightness necessary for efficient movement.
But if that balance tips, it can contribute to the fragility seen in osteoporosis, proving that even a chemical associated with well-being can be detrimental in excess.
This theme of delicate moderation continues into the soft, vital organs that keep us alive. In the liver, serotonin helps regulate blood flow and creates the conditions needed for cellular regeneration after injury. In the pancreas, it modulates the release of insulin, tying our mood regulator directly to our blood sugar and metabolism. However, the most critical example of this balancing act is found in the heart. The valves that open and close with every beat are lined with cells highly sensitive to serotonin. If the bloodstream carries a chronically high load of this chemical, perhaps due to a rare tumor or the side effects of certain drugs, it can trigger a process called cardiac fibrosis. The serotonin overstimulates the production of collagen, causing the flexible, fluttering leaflets of the heart valves to thicken and stiffen. They lose their ability to seal tightly, becoming rigid like scar tissue. It is a stark illustration of how a substance essential for healing wounds can, if left unchecked, overheal the body into a state of rigidity. The system relies not just on the presence of this messenger, but on its precise rhythmic control to ensure that our tissues remain pliable and our bones remain resilient. To find the source of this rhythm, we must travel upward from the spine and enter the most ancient, fundamental architecture of the skull. Deep within the brainstem, that primitive stalk connecting our physical body to the vast complexities of the mind, lies the primary manufacturing plant for the brain's supply of serotonin. This region is known as the Raphe nuclei. The name itself is a subtle map of the terrain. Raphe is derived from the Greek word for a seam or a suture. It is a fitting designation as these clusters of neurons are situated almost exactly along the midline of the brainstem, stitching together the left and right sides of this vital column like the central seam running up the back of a heavy velvet curtain. What is perhaps most remarkable about this system is the sheer scale of disparity. The human brain is a galaxy of nearly 100 billion neurons, a number so vast it is difficult to comprehend. Yet the cells that comprise the Raphe nuclei, the cells responsible for producing almost all of the serotonin that regulates our mood, sleep, and perception, number only in the tens of thousands. They are a tiny, elite minority huddled in the basement of the brain. If we were to judge them solely by their population size, they would seem insignificant, yet their influence is absolute. They act as the central modulation station for the entire nervous system, proving that in biology, location and connectivity often matter far more than sheer quantity. From these few concentrated clusters in the brainstem, a miracle of biological engineering unfolds. The neurons here do not simply talk to their neighbors, they project long, winding cables called axons that travel immense distances to virtually every corner of the brain. Imagine a great ancient tree with its roots buried deep in the soil of the brainstem, but its canopy spreading so wide that it shades the entire landscape. This is the architecture of the serotonin system. The axons originating in the Raphe nuclei branch out profusely, creating a dense, intricate lattice that permeates the cerebral cortex, the cerebellum, and the spinal cord. The distribution method is often compared to a sprinkler system or a misting fountain. Unlike other neural circuits that send precise point-to-point messages like a telephone call, serotonin neurons often release their chemical payload from swellings along their axons called varicosities. These varicosities act like beads on a string, leaking serotonin into the spaces between cells, allowing the chemical to diffuse through the tissue and bathe large networks of neurons simultaneously. This specific geography means that a single neuron in the Raphe nuclei can have an axon that branches so widely it influences hundreds of thousands of targets across completely different regions. The dorsal Raphe nucleus, the largest of these clusters, sends its projections sweeping forward into the frontal cortex and the basal ganglia, touching the areas responsible for movement, planning, and complex thought. Meanwhile, the median Raphe nucleus tends to weave its connections into the hippocampus, the seat of memory and spatial navigation. Through this expansive fountain-like wiring, the brain ensures that serotonin is not just a local messenger, but a global state setter, capable of shifting the entire climate of the mind from high alert to a calm, unified focus. To achieve such a sweeping transformation, serotonin cannot simply shout a single command to every cell it touches. Instead, it utilizes a sophisticated system of translation, a biological language where the meaning of the message depends entirely on who is listening. This is where we encounter the extraordinary diversity of the serotonin receptor family, a collection of protein structures that act as the gatekeepers of cellular function. In the microscopic landscape of the neuron, serotonin is the universal key, precise and unchanging in its chemical shape.
However, the locks it fits into, known scientifically as the 5-HT receptors, vary wildly in their internal mechanisms and intended effects. When serotonin enters one type of lock, it might turn the mechanism to the left, effectively shutting a door and quieting the cell. When it enters a different lock on a neighbor cell, it might turn the mechanism to the right, throwing the door wide open and sparking a burst of electrical activity. This variety is categorized into 7 distinct families, numbered 5-HT1 through 5-HT7, a classification system that barely scratches the surface of the underlying complexity. Within these 7 families, scientists have identified at least 14 distinct receptor types, each one distinct enough to radically alter the outcome of the chemical conversation. Consider the profound contrast between the 5-HT1 and 5-HT2 families. Receptors in the 5-HT1 group generally act as inhibitors. When serotonin binds to them, it essentially tells the neuron to relax, to reduce its firing rate, and to dampen the noise. This is the mechanism often associated with the relief of anxiety, a cellular hush that spreads through the neural networks like a blanket of fresh snow. Conversely, the 5-HT2 receptors often serve as excitors. When the key turns in these locks, the neuron becomes more likely to fire, driving wakefulness, or, when stimulated excessively, contributing to the vivid intensity of hallucinations. This duality explains how a single molecule can manage such contradictory states as sleep and wakefulness, or nausea and appetite. It is not the molecule that changes, but the reception. It is reminiscent of an orchestra where the conductor raises a baton, the serotonin, and the violin section plays a soft, mournful note, while the percussion section strikes a thunderous beat. Both sections saw the same signal, but their instruments, the receptors, translated it into entirely different sounds. The body requires this nuance because a blunt instrument would be dangerous. If serotonin simply turned everything on, the result would be a storm of uncoordinated activity. And if it turned everything off, the system would collapse into stasis. Among these families, there are even differences in speed. Most serotonin receptors are what we call G-protein coupled receptors, which means they work somewhat slowly, triggering a cascade of internal changes that might take seconds or minutes to fully manifest. They are the slow architects of mood and personality. However, the 5-HT3 receptor is the exception, the rebel of the family. It is an ion channel, meaning that when serotonin attaches, it opens a physical tunnel in the membrane almost instantly, allowing charged particles to rush in. This is the speed required for rapid reflexes, governing fast biological responses like the immediate sensation of nausea if we ingest something toxic. Furthermore, some of these receptors function as sensors for the serotonin system itself. Located on the sending neuron, they act like a thermostat, detecting how much serotonin is floating in the synapse. If the levels get too high, these specific receptors send a signal to stop production, a feedback loop that maintains a delicate equilibrium. The sheer number of these receptor types means that serotonin is never just one thing. It is a shapeshifter, defined less by what it is and more by where it lands.
This receptor diversity is the fundamental reason why targeting serotonin in medicine is so complex, as hitting the wrong receptor can trigger a side effect just as easily as the right receptor triggers a cure, creating a chemical high-wire act that occurs billions of times every second within the quiet cathedral of the skull. When this delicate chemical balancing act is successful, the result is not an explosion of euphoria or a manic burst of energy, but rather the subtle, often unnoticed sensation of simply being okay. In the popular imagination, serotonin is frequently mislabeled as a happy chemical, suggesting that more of it equals more joy, much like turning up the volume on a radio. However, the reality of its function is closer to a master regulator, a stabilizer that keeps the emotional landscape within a livable, sustainable range, often referred to by neuroscientists as the Goldilocks zone of mood. Not too high, not too low, but just right for navigating the complexities of the human experience. To truly understand the specific flavor of well-being that serotonin provides, it is helpful to contrast it with its more famous and often louder counterpart, dopamine. If the brain were a grand orchestra, dopamine would be the brass section, blasting the triumphant notes of reward, anticipation, and the thrill of the chase. Dopamine is the neurochemical driver of I want. It is the molecule of motivation that pushes an animal to hunt, to seek a mate, or to scroll to the next video. It provides the spike of pleasure when a goal is achieved, but that spike is inherently transient, designed to fade quickly so that the pursuit can begin again. Serotonin, conversely, operates as the conductor of the strings and woodwinds, creating the underlying harmony that allows the melody to exist. It is the molecule of I have. While dopamine is about the acquisition of resources, serotonin is the chemical signal that acknowledges those resources are now secure. It is the physiological manifestation of satiety and safety.
When serotonin levels are optimal, the brain settles into a state of contentment where the urge to constantly seek more is quieted. This is why low serotonin is often linked not just to sadness, but to a restless, gnawing sense of dissatisfaction, a feeling that something is missing even when one's surroundings are perfectly adequate. This stabilizing influence extends deep into the architecture of emotional processing. One of serotonin's primary roles is to regulate the sensitivity of the amygdala, the almond-shaped structure deep in the temporal lobes responsible for detecting threats and processing fear. You might think of serotonin as the brain's diplomatic corps, negotiating with the amygdala to prevent it from declaring war over minor infractions. When serotonin activity is robust, the connection between the emotional centers and the rational executive centers of the prefrontal cortex is strengthened. This allows for what psychologists call top-down processing, where the logical mind can gently veto a sudden surge of anxiety or irritation before it cascades into a full-blown reaction. In this way, serotonin functions as a filter for negative emotional processing. It raises the threshold for what the brain considers an emergency. With sufficient serotonin, a rude comment from a stranger or a minor traffic delay is processed as a trivial nuisance, easily brushed aside as you continue with your day. Without that chemical buffer, the exact same external event can trigger a disproportionate internal crisis, spiraling into rage or despair. The world hasn't changed, but the chemical lens through which it is viewed has lost its protective coating. Therefore, if we were to define happiness in strictly neurochemical terms based on this system, it would not be the presence of constant ecstasy, but the absence of unnecessary suffering. It is the quiet confidence that the current moment is safe, that the self is capable, and that the immediate future is manageable. It is the background hum of resilience that allows a person to wake up, look at the ceiling, and feel that the day ahead is something they can handle, a state of being that is less about the peaks of pleasure and more about the expansive, peaceful plateaus of peace.
This internal quiet allows for something remarkable to happen within the physical architecture of the brain. We often imagine memory as a static archive, a dusty library where books are stored and retrieved. But the reality is that your memory is a living, shifting landscape, and serotonin is its primary gardener. Deep inside the temporal lobes resides the hippocampus, a curved seahorse-shaped structure that acts as the gateway for new memories. It is here that serotonin performs one of its most impressive feats, the regulation of neuroplasticity. For a long time, science held on to the belief that the adult brain was fixed, unable to grow new cells. We now know this is untrue, thanks to the discovery of neurogenesis, the birth of new neurons. Serotonin is a critical catalyst in this process. It modulates the release of brain-derived neurotrophic factor, often abbreviated as BDNF. You can think of BDNF as a sort of biological fertilizer. When serotonin signaling is healthy, it encourages the brain to sprout new connections and strengthen existing ones, particularly in the hippocampus. This means that the chemical responsible for stabilizing your mood is directly responsible for the physical expansion of your mind's capabilities. This cellular growth translates into the practical ability to learn. When you are trying to master a new skill, whether it is playing an instrument or learning a new language, serotonin is working behind the scenes to help encode these experiences into long-term storage. It facilitates the synaptic changes that make learning feel fluid rather than strenuous. Conversely, when serotonin levels dip, we experience the inverse of this fluidity. We call it brain fog, but it is technically a form of cognitive rigidity. Without enough serotonin to grease the neural gears, the brain loses its flexibility. A mind low in serotonin often struggles to shift attention or adapt to changing circumstances. You might find yourself stuck in a loop of repetitive thoughts, unable to see a solution that is right in front of you, or reading the same sentence 3 times without absorbing its meaning. This happens because serotonin functions as a gain control system for your neurons. It dampens the background noise of irrelevant neural activity so that the important signals, the things you actually want to focus on, can come through with clarity. It essentially improves the signal-to-noise ratio of your consciousness. Therefore, the presence of this molecule does more than just make you feel content, it makes you sharper. It creates the biological conditions necessary for adaptability. In a very real sense, your ability to change your mind, to update your beliefs based on new information, and to rewrite the narrative of your life depends on the presence of this neurotransmitter. It grants you the cognitive flexibility to navigate a complex world without breaking under the pressure, turning the chaos of daily sensory input into an organized, coherent stream of thought. This capacity for organization is directly linked to one of the most vital functions of the adult brain, the ability to wait. While we often think of patience as a moral virtue or a practiced discipline, neurobiology reveals that it is fundamentally a chemical capability, heavily reliant on serotonin transmission. The brain is constantly weighing the value of immediate rewards against the promise of future gains, a calculation known in psychology as temporal discounting. Serotonin is the primary modulator of this scale. When levels are optimized, the brain can accurately value the future, allowing you to tolerate the passage of time without agitation. This mechanism is essentially a behavioral brake. It inhibits the impulse to act on every fleeting desire or frustration, creating a necessary gap between feeling and reaction. Without this neurochemical buffer, the now becomes overwhelming. This phenomenon has been starkly illustrated in clinical research involving tryptophan depletion. Since the body cannot synthesize serotonin without the amino acid tryptophan, scientists can temporarily lower serotonin levels in study participants by restricting this specific nutrient in their diet.
The results of such studies are consistent and revealing. When serotonin availability drops, individuals who are normally prudent and careful suddenly begin to exhibit impulsive decision-making. In laboratory gambling tasks, for instance, participants with temporarily lowered serotonin become far more likely to choose small, immediate monetary rewards rather than waiting for larger payouts that would be available after a short delay. The neurological cost of waiting becomes too high for the brain to bear. The future, in a low serotonin state, feels abstract and uncertain, while the present impulse feels absolute. This correlation extends beyond simple patience into the realm of risk and aggression. The waiting mechanism is part of a broader system of behavioral inhibition. In the wild, an animal that cannot wait for the opportune moment to strike, or one that cannot suppress the urge to fight a superior opponent, is unlikely to survive. Evolution has utilized serotonin to regulate this stillness. It allows a predator to remain motionless for hours, suppressing the urge to move until the probability of success is highest. In humans, this translates to social prudence and risk management. Low serotonergic activity is frequently associated with high-risk behaviors and a lack of premeditation. The impulse to act becomes a short circuit, bypassing the prefrontal cortex, where logical planning resides. Conversely, when serotonin is functioning correctly, it provides the cool temperament required to assess danger and delay gratification. It transforms the agonizing sensation of waiting into a manageable state of anticipation. It quiets the neural noise that demands instant resolution, allowing you to sit with uncertainty. In this light, serotonin is not just a molecule of happiness, but the molecule of endurance, granting us the profound ability to invest in a tomorrow that has not yet arrived. To reach that tomorrow, however, the brain must first navigate the immediate threats of today, a task managed by the ancient sentinel known as the amygdala. Buried deep within the temporal lobes, this almond-shaped cluster of nuclei is responsible for processing fear and detecting danger, operating at a speed that often bypasses conscious thought entirely. When the amygdala perceives a threat, whether it is a predator in the grass or a stressful email in your inbox, it prepares to sound the alarm that triggers the fight or flight response. Serotonin acts as the primary chemical brake on this alarm system. It functions as an inhibitory neurotransmitter within the amygdala, binding to specific receptors to dampen the electrical firing of neurons that would otherwise scream panic. Without sufficient serotonin activity in this region, the amygdala becomes hyperactive, interpreting harmless shadows as monsters and minor inconveniences as catastrophes. This is the biological basis of anxiety, a security system that cannot be turned off because the chemical key is missing. When serotonin is present in the correct quantities, it raises the threshold for what the brain considers a crisis. It allows the amygdala to distinguish between a genuine survival threat and a mere moment of uncertainty, preventing the cascade of adrenaline and cortisol that wreaks havoc on the body during prolonged stress. Before we explore how the brain physically unlearns these fear responses, I'm curious to know what personal rituals you rely on to signal safety to your own mind. Do you find peace in deep breathing, the sound of rain, or perhaps simply in learning about the quiet mechanics of your own biology? Feel free to share your methods for finding calm in the comments. This subjective feeling of calm you experience is actually the result of serotonin strengthening the communication lines between the emotional brain and the rational brain. Specifically, serotonin facilitates the connection between the amygdala and the prefrontal cortex, the center of logic and planning. Under normal circumstances, the prefrontal cortex acts as a parent to the amygdala, telling it to stand down when a fear is irrational. Serotonin provides the bandwidth for this conversation to happen. When serotonin levels drop, this connection frays, and the rational brain loses its ability to soothe the emotional brain, leaving the individual at the mercy of raw, unfiltered instinct. Furthermore, serotonin is essential for a process known as fear extinction, which is the brain's ability to learn that a previously threatening situation is now safe. If you have ever been afraid of a specific place because of a bad memory, but then gradually felt comfortable there again after repeated safe visits, that is fear extinction in action. This unlearning of fear relies heavily on plastic changes in the neural circuits of the amygdala, changes that are mediated by serotonin signaling. It transforms the world from a landscape of potential dangers into a manageable environment, physically altering the way nerve cells fire to ensure that the memory of fear does not become a permanent state of being. And yet, this calming influence over our fears is only half of the story, serving as the daylight act in a 24-hour cycle of biological stewardship. When the sun begins to dip below the horizon and the light in our environment fades, serotonin prepares for its second, perhaps even more profound performance. It is a transformation that highlights the remarkable efficiency of the human body, for the molecule that keeps us awake, alert, and happy is the exact same raw material the body uses to construct the molecule of sleep. This is one of the most elegant facts of human biology. Serotonin is the direct chemical precursor to melatonin. Deep within the center of the brain lies the pineal gland, a tiny pine cone-shaped structure often referred to historically as the seat of the soul, but scientifically known as the body's master clock tower. During the daylight hours, the pineal gland is relatively quiet, as the presence of blue light striking the retina sends an inhibitory signal that prevents it from working. However, as darkness falls, that brake is released and the pineal gland begins to draw in the serotonin that has been circulating in the brain all day. It does not create the hormone of sleep from scratch. Instead, it acts as a molecular alchemist, recycling the serotonin of the day into the sedative of the night. The process is a precise two-step enzymatic dance. First, an enzyme with the complex name of aryl alkylamine N-acetyltransferase, often simply called the TYMEzyme by chronobiologists, attaches a small chemical cluster known as an acetyl group to the serotonin molecule. This simple addition fundamentally changes the molecule's shape and function, turning it into an intermediate substance called N-acetylserotonin. This intermediate compound doesn't linger for long. A second enzyme immediately performs a process called methylation, adding a methyl group to the structure. With this final adjustment, the transformation is complete. The alertness of serotonin has been chemically folded away, and the molecule is now melatonin. This pathway explains why the two chemicals are so structurally similar, often described as chemical cousins, though chemical siblings might be more accurate. It also sheds light on why dietary tryptophan, the amino acid needed to make serotonin, is often linked to better sleep. By increasing the available serotonin, you're essentially stocking the shelves of the factory that produces melatonin. Without sufficient serotonin during the day, the pineal gland lacks the necessary bricks to build the hormone of darkness at night. This biological recycling program suggests that a healthy, happy wakefulness is a prerequisite for deep, restorative sleep, as the chemical intensity of the day is literally converted into the chemical tranquility of the night. This conversion is strictly regulated by light, which is why modern screens can be so disruptive. The blue light emitted by phones and televisions mimic the wavelength of the noon sun, tricking the suprachiasmatic nucleus, the pacemaker of the brain, into believing it is still daytime. This signals the pineal gland to halt the conversion process, leaving the brain awash in serotonin and N-acetylserotonin, but starving it of the melatonin required to initiate the sleep cycle. It is a fragile system, evolved over millions of years of reliable sunsets, now navigating a world of artificial luminescence. When the conversion works correctly, the rise in melatonin doesn't simply knock you out like a sedative drug might. Instead, it acts as a chemical signal, a master switch that tells the body's internal organs that the business day is over.
It lowers core body temperature, reduces blood pressure, and quiets the chatter of the nervous system. It is interesting to consider if you have ever noticed that specific wave of heaviness that hits you when you finally turn off the last lamp, realizing just how much light was holding back the tide of sleep. That sensation is the physical feeling of the serotonin-melatonin pathway finally being allowed to flow without obstruction. The efficiency of this system is paramount because melatonin has a very short half-life, meaning it breaks down quickly. The body must constantly produce it throughout the night to maintain sleep, requiring a steady supply of serotonin from the reserves built up during the day. This dependency creates a cyclical harmony where the quality of your day dictates the quality of your night, and the restfulness of your night replenishes the serotonin potential for the following morning. It is a closed loop of transmission, ensuring that the biology of waking and the biology of sleeping are not separate systems, but two phases of the same molecular rhythm.
This molecular rhythm is tied inextricably to the rotation of the Earth, anchored by the most powerful signal in our environment, which is sunlight. When the very first photons of morning light strike the retina, they bypass the visual cortex, the part of the brain that processes images, and instead strike a specialized group of cells called intrinsically photosensitive retinal ganglion cells. These cells are not there to help you see the world. They are there to tell your biology what time it is. Upon detecting the specific blue light frequency of dawn, they send an immediate electrical signal to the suprachiasmatic nucleus, the master clock, which in turn signals the Raphe nuclei to ramp up the production and release of serotonin. It is a direct translation of electromagnetic energy into chemical alertness, effectively jump-starting the engine of consciousness. This explains why serotonin levels naturally track the arc of the sun, rising sharply at dawn, peaking during the intense brightness of midday, and beginning to taper off as the light fades in the evening. It acts as the chemical marker of the active phase, signaling to every cell in the body that this is the time to be awake, to move, and to engage with the world. However, this system relies on a level of brightness that modern indoor living rarely provides. Outdoor sunlight, even on a cloudy day, is thousands of times brighter than the artificial bulbs in our homes and offices. When we spend our days in dim interiors, we are effectively living in a state of biological darkness, resulting in a blunted serotonin peak that leaves the brain hovering in a twilight zone of low energy. This dependency on light becomes most apparent during the shift of seasons. As the days shorten and the angle of the sun drops in winter, the reduced duration and intensity of light hitting the eye leads to a measurable decline in serotonin activity. This is the primary biological mechanism behind seasonal affective disorder, where the brain is essentially starved of the light cues it needs to synthesize enough of this vital neurotransmitter. Evolutionarily, this drop in serotonin might have encouraged our ancestors to conserve energy and sleep more during scarce winter months, but in the modern world, it manifests as a persistent mood slump. Interestingly, the density of serotonin transporter proteins, the molecular trucks that clear serotonin out of the synaptic gap, also fluctuates with the seasons. Research suggests these transporters are more active in winter, sweeping away the little serotonin available more aggressively, which exacerbates the shortage. This seasonal ebb and flow highlights just how porous the barrier is between our internal chemistry and the external world. Perhaps the most critical aspect of this light-serotonin relationship is its impact on the night that follows. There is a profound chemical paradox at play here. To sleep deeply in the dark, you must first be exposed to bright light.
This is because serotonin is the direct chemical precursor to melatonin, the hormone of darkness. The brain uses the serotonin you build up during the day as the raw material to synthesize melatonin once the sun goes down. If your daytime serotonin peak is weak due to a lack of morning light, your nightly melatonin production will be correspondingly low. You are essentially building the chemical pressure for sleep the moment you step outside in the morning, creating a reservoir of potential rest that will only be unlocked hours later. The brighter the morning, the deeper the eventual darkness feels to the brain, closing the loop between the wakeful sun and the restorative night. Just as the light dictates the rhythm of your wakefulness, internal chemical tides dictate the rhythm of your appetite. This same biological architecture that tracks the movement of the sun also monitors the fuel reserves within your system, centering its complex operations deep inside the hypothalamus. Here, serotonin takes on a role quite different from its famous job in mood regulation, functioning instead as the body's primary essential signal for satiety. It acts as the chemical equivalent of a heavy, satisfied sigh after a nourishing meal, turning down the volume on the primal drive to consume. To understand this quiet power, we must look at the loud voice it opposes. There are hormones like ghrelin which actively shout at the brain to demand energy, creating that gnawing, urgent sensation of an empty stomach. Ghrelin is the alarm bell of the metabolic system. Serotonin, by contrast, is the gentle hand that reaches out to silence that alarm. When you consume food, and as the mechanical stretching of the stomach combines with rising nutrient levels in the blood, serotonin neurons begin to fire rapidly. They flood specific synaptic gaps to deliver a clear, unmistakable message: the need has been met. This signaling process is incredibly specific, relying on precise molecular geometry. Scientists have identified a particular docking station known as the 5-HT2C receptor, which acts as the dedicated lock for this fullness key. When serotonin binds here, it actively suppresses the neural circuits that drive feeding behavior. It effectively switches the brain from acquisition mode, where you are biologically programmed to hunt and gather energy, to preservation mode, where the body can rest, digest, and allocate resources to repair. This interaction is so fundamental to our biology that medications designed to assist with weight management often target this exact receptor, attempting to replicate the natural chemical message that the tank is full. There is a fascinating, almost manipulative nuance to this system regarding the specific types of food you crave. You may have noticed that during moments of stress or low mood, the body rarely craves kale or lean protein, it craves carbohydrates. This is not a failure of willpower, but a sophisticated biochemical maneuver orchestrated by the brain. Carbohydrate-rich foods trigger the release of insulin, which acts like a traffic controller in the bloodstream. Insulin clears most competing amino acids from the bloodstream, sweeping them into muscle cells, but it leaves tryptophan, the raw material for serotonin, behind. With the competition removed, tryptophan has a wide, clear path to cross the blood-brain barrier. The result is a rapid, efficient spike in serotonin production. Essentially, when you reach for comfort food, you are unconsciously manipulating your own neurochemistry to engineer a sense of calm and fullness. The dance between hunger and satiety is a constant, delicate negotiation. If serotonin levels are chronically low, or if the receptors become less sensitive, the stop signal becomes faint, like a whisper in a noisy room. The brain may never quite receive the authoritative message that the body is satisfied, leading to a perpetual state of searching for food. Conversely, when the system is balanced, the transition from hunger to fullness happens seamlessly. It is a sensory loop where the physical act of eating translates into a chemical state of peace. This mechanism ensures that an organism doesn't just eat endlessly, but pauses to utilize the energy it has gathered. It prevents us from becoming overly weighed down, maintaining a balance between fueling the machine and keeping it light enough to move. The feeling of being full is, at its core, a serotoninergic event, a successful communication between the gut and the brain that survival has been secured, allowing the mind to drift away from the hunt and settle into safety. But that drift into safety often requires a specific type of fuel to initiate the chemical cascade. You might have noticed that when stress rises or mood dips, the body rarely cries out for celery or plain grilled chicken. Instead, there is a distinct magnetic pull toward carbohydrates, warm bread, heavy pasta, potatoes, and sweets. This specific hunger is not a failure of willpower, nor is it a simple desire for taste. It is a calculated request from the brain's serotonin system, a biological demand for the raw materials needed to manufacture peace. To understand this, we have to look at the blood-brain barrier, the selective border that strictly controls what enters the central nervous system. Tryptophan, the amino acid precursor required to build serotonin, faces a significant logistical challenge at this border. It must compete for transport with other large neutral amino acids, such as leucine, valine, and tyrosine, which are typically more abundant in the bloodstream. Under normal conditions, tryptophan is simply crowded out, struggling to cross into the brain in significant quantities because the transport channels are clogged with these other molecules. This is where the carbohydrate craving serves a distinct evolutionary purpose. When you consume carbohydrates, your pancreas releases insulin to manage the glucose. Interestingly, insulin has a secondary effect that is crucial for your emotional state. It drives those competing amino acids out of the bloodstream and into your muscle tissues, but it leaves tryptophan largely untouched. By clearing away the competition, insulin essentially opens a VIP lane on the highway, allowing tryptophan to travel freely across the blood-brain barrier without resistance. Once inside the brain's sanctuary, levels of tryptophan rise and serotonin production is immediately ramped up. This is the physiological basis of what we often dismiss as emotional eating. That bowl of macaroni or slice of toast is effectively a form of unconscious self-medication, a rapid, instinctual way to alter brain chemistry and induce a state of calm. It explains why we reach for starchy foods during heartbreaks, stressful work weeks, or the darker months of the year. The resulting feeling of sedation is not accidental. It is the somatic experience of the brain successfully synthesizing the molecule of satisfaction, turning a simple meal into a chemical sigh of relief. Yet to think of this molecule solely as a provider of human comfort is to ignore the vast majority of its history. This chemical sigh of relief is actually an ancient biological shout that has echoed across the planet for billions of years, long before the first human brain ever pondered the concept of happiness. Serotonin is arguably one of evolution's most successful inventions, a primordial signaling tool that life adopted almost as soon as it began to flourish. It is found in simple single-celled organisms, in fungi, and in the green silence of the forests, suggesting that its original purpose had nothing to do with mood and everything to do with basic survival. If you were to analyze the chemical composition of a black walnut, you would find it teeming with serotonin.
The same is true for bananas, plantains, pineapples, tomatoes, plums, and even kiwis. In fact, plants produce huge quantities of the stuff, but a banana does not feel joy, nor does a walnut suffer from anxiety. In the vegetable kingdom, this molecule serves a much more utilitarian purpose, acting as a potent growth regulator. It helps organize the architecture of root systems, telling the plant how to anchor itself in the soil,
And it assists in the reproductive process, signaling tissues to mature. In many fruits, serotonin acts as a powerful antioxidant, scavenging for dangerous free radicals and protecting the precious genetic cargo inside the seeds from rotting before they have a chance to sprout. It is a preservative, a guardian of the future generation. However, nature is rarely sentimental, and it is known for repurposing its tools in surprising ways. The same compound that stabilizes your mood and protects a walnut seed is also weaponized by other species to inflict immediate, sharp distress. Consider the stinging nettle. When you brush your leg against its leaves and feel that sudden burning itch, you are experiencing a direct injection of serotonin mixed with histamine and acetylcholine. In this context, it is not a soothing balm, but a chemical alarm. The plant uses it to overstimulate the nerve endings of grazing animals, teaching them a painful lesson about what not to eat. This strategy is not limited to flora. The venom of the paper wasp, the hornet, and certain species of scorpions is laced with serotonin. In these venomous cocktails, the molecule acts as a vasoconstrictor and a pain amplifier. It causes blood vessels to tighten and pain receptors to flare, ensuring that the venom spreads effectively and that the predator remembers the encounter vividly. This incredible versatility highlights the supreme efficiency of evolution. Nature does not reinvent the wheel for every new species. It simply repurposes the tools lying around the genetic workshop. A molecule used for defense in a nettle became a molecule of digestion in a worm, which eventually became a molecule of consciousness in a human. It suggests that before serotonin was ever a regulator of emotion, it was simply a fundamental instruction, a chemical note that told a living cell to pay attention, to grow, to defend, or to endure. That chemical note still resonates in the deepest, darkest corners of the ocean floor, where creatures that separated from our own evolutionary tree more than 350 million years ago still use it to organize their entire existence. The common lobster has become quite famous in neurochemistry circles, not for its culinary value, but because its nervous system is large, accessible, and remarkably similar to our own in how it handles the concept of status. Lobsters live in a harsh environment where the best hiding spots and the richest food sources are scarce, and to survive, they must establish a pecking order. This is not a polite social arrangement, but a rigid dominance hierarchy determined by physical confrontation. When two lobsters meet, they engage in a ritualized dance of aggression, measuring each other's size, claw strength, and willingness to fight. It is here, in the midst of this underwater standoff, that serotonin dictates the outcome. Biologists have discovered that a lobster with high levels of serotonin will assume a posture of dominance. It stands tall on its legs, raises its claws, and essentially flexes its entire body to appear as large and intimidating as possible. This serotonin-fueled posture is not merely a bluff. The chemical actually primes the lobster's neural circuitry for sustained effort and aggression. It makes the creature brave. It suppresses the urge to flee and heightens the reflexes required for combat. The lobster with the most serotonin flowing through its system is statistically much more likely to win the fight, secure the shelter, and claim the territory. Conversely, the loser of these confrontations experiences a chemical crash that is the biological opposite of confidence. In the crustacean brain, the counterbalance to serotonin is a neurochemical called octopine. While high serotonin correlates with the rigid, expansive posture of the victor, high octopine correlates with the slumping, curl-up posture of the defeated. A defeated lobster will tuck its tail, lower its claws, and retreat backwards, signaling to the world that it is no longer a threat. This is a survival mechanism, ensuring that the weaker animal does not continue a fight it cannot win, which would likely result in death. What is truly fascinating is how easily this chemical balance can be manipulated, proving that status is often just a matter of molecules. Researchers have found that if you take a defeated, sulking lobster, one that has just lost a fight and is full of octopine, and inject it with serotonin, it will immediately stretch out its limbs. It will turn around and march back towards the dominant lobster that just beat it, ready to fight again, often fighting longer and harder than it did the first time. The memory of defeat is chemically overwritten by the molecule of dominance. Even more striking is that antidepressants designed for humans, which work by increasing the availability of serotonin in the brain, have the exact same effect on lobsters.
If you dissolve selective serotonin reuptake inhibitors in the tank water, even the most submissive, low-ranking lobsters will begin to posture like alphas. This suggests that the biological machinery for confidence and social standing is incredibly ancient. Long before humans developed language, culture, or complex societies, nature had already devised a chemical system to tell an organism where it stood in relation to its peers. This deep evolutionary root implies that the feeling of confidence and the physical stance that accompanies it is not a modern psychological invention. When we stand up straight, pull our shoulders back, and face the world, we are tapping into a neural circuit that has been operating on this planet for hundreds of millions of years. The serotonin system acts as a kind of internal status tracker, constantly monitoring our position in the social world and adjusting our biology accordingly. It tells the organism whether it is safe to expand and take up space, or whether it is necessary to shrink and hide. In the lobster, this regulation is absolute and mechanical, determining who gets the best burrow and who is left exposed. In humans, this system is infinitely more complex, interwoven with our thoughts, our memories, and our subtle social interactions. Yet the fundamental signal remains the same. Have you ever noticed how your own physical posture shifts instinctively? How you might slump after a disappointment or stand taller after a success without you ever consciously telling your muscles to move? This automatic alignment of the body with the mind is the work of serotonin, bridging the gap between our internal state and our external reality. It suggests that our perceptions of status and self-worth are not just abstract concepts floating in the mind, but deep physiological states regulated by the same ancient chemistry that governs the aggressive posturing of crustaceans on the ocean floor. The molecule that once told a worm to move toward food eventually evolved to tell a lobster to stand its ground, and later to tell a human being that they are respected and secure. When that chemical signal of security is absent, however, the ancient machinery of the brain defaults to a much more volatile state. One of the most consistent findings in behavioral neuroscience is the inverse relationship between serotonin levels and aggression. Across a vast spectrum of species, from the humble fruit fly to the complex human being, low levels of serotonin activity are strongly correlated with an increase in hostility, irritability, and impulsive violence. You might think of this neurotransmitter as the brain's primary cooling fluid. Deep within the brain lies the amygdala, a small almond-shaped structure responsible for processing threats and generating fear or anger. It is the engine of our primal survival instincts, always ready to react to danger. In a well-regulated brain, the prefrontal cortex, the logical, modern part of the mind, acts as a supervisor, sending inhibitory signals to calm the amygdala down. Serotonin is the essential messenger that carries these calming orders. When serotonin levels are optimized, the brakes work effectively.
You might feel a flash of annoyance when someone cuts you off in traffic, but you remain in control. The impulse is dampened before it becomes an action. However, when serotonin turnover is low, that braking mechanism fails. The line between feeling an emotion and acting on it becomes dangerously thin. This specific type of aggression is known as impulsive aggression. It is not the calculated, cold aggression of a chess player planning a trap, but rather the explosive, reactive aggression of a short fuse burning down in an instant. Scientists have measured the breakdown products of serotonin in the cerebrospinal fluid of various mammals and found that lower concentrations of these metabolites almost always predict a higher likelihood of risk-taking and physical confrontation. This biological reality suggests that what we often call civilization or manners is, at a chemical level, a function of serotonin availability. It is the molecule of cooperation. For social animals to live together in groups, they must be able to tolerate minor provocations without resorting to lethal combat. Every time you choose to let a grievance go or choose to share resources instead of hoarding them, you are relying on the subtle influence of this neurotransmitter to override your selfish or defensive instincts. In this light, serotonin does far more than just regulate mood. It actively facilitates the social contract. It suppresses the urge to harm others and enhances the neural pathways associated with grooming, nesting, and bonding. It shifts the brain's bias away from the paranoid defense of territory and towards the collaborative safety of the group. Kindness, patience, and tolerance are not just abstract moral virtues, but tangible biological states supported by a steady flow of this specific chemical. When the brain is awash in serotonin, the world feels less like a battleground and more like a community, allowing us to perceive the intentions of others with less suspicion and more empathy. This biological foundation of empathy naturally extends into the complex realm of human connection, where serotonin performs an intricate dance with other powerful neurotransmitters like dopamine and oxytocin.
In the grand orchestration of social bonding, serotonin acts as a steadying hand, modulating the intensity of our emotional responses to ensure that connections are safe and sustainable. However, one of the most counterintuitive facts about the chemistry of romance is that falling in love usually causes serotonin levels to plummet rather than rise. During the initial phase of intense infatuation, often called the honeymoon period, the brain's serotonin drops to levels surprisingly similar to those found in individuals diagnosed with obsessive-compulsive disorder. This temporary depletion is likely what drives the obsessive nature of new love, creating a state where you simply cannot stop thinking about the other person. It creates a laser-like focus, suppressing the biological caution we usually feel toward strangers and allowing us to take the emotional risk of bonding. There is a distinct chemical seesaw effect at play here. As serotonin lowers, dopamine, the chemical associated with craving and motivation, spikes, creating the giddy, high-energy state of early romance. But the body cannot sustain this high-alert metabolical emergency forever. As a relationship matures and deepens into long-term attachment, typically after the first 12 to 18 months, serotonin levels gradually rise back to their baseline. This restoration brings a sense of calm, stability, and comfort, replacing the frantic energy of infatuation with the secure, soothing bond of companionate love. In this stable phase, serotonin works closely with oxytocin, often called the bonding hormone, to reinforce social reward. Specifically, within the nucleus accumbens, the release of serotonin stimulates the release of oxytocin, making physical touch and proximity feel deeply soothing rather than just electrifying.
This interaction is not limited to romantic partners, as it is the same chemical framework that underpins deep friendships and familial bonds. While that feeling of calm connection is universal, the biological machinery that regulates it varies significantly from person to person, hidden deep within your genetic code. This brings us to the fascinating mechanics of the serotonin transporter protein, often referred to by scientists as SERT. You might think of this protein as a diligent recycling manager operating within the brain's synaptic gaps. When a message is sent between neurons, serotonin is released to bridge the divide, but it cannot stay there forever. The transporter protein's job is to sweep the serotonin back up into the cell to be reused or broken down, a process known as reuptake, which effectively turns off the signal and resets the system for the next message. The instructions for building this recycling system are written in a specific gene known as SLC6A4. But what makes this area of study so compelling is a region called 5-HT-TLPR. In the human population, this genetic sequence appears in two primary variations, a long form and a short form. The long form allows the brain to build these transporters efficiently, creating a robust recycling system that clears serotonin quickly. However, the short form is less efficient, resulting in fewer transporters and a different chemical rhythm within the brain. If you possess one or two copies of the short allele, your neural landscape operates with a heightened level of sensitivity. Research suggests that individuals with the short variation of this gene tend to have a more reactive amygdala, the part of the brain responsible for processing emotions and detecting threats. This does not mean the system is broken, but rather that it is tuned to a higher frequency of awareness. In evolutionary terms, this heightened sensitivity was likely an asset, allowing certain members of a tribe to detect danger earlier than others. In modern psychology, this genetic difference is often used to explain why some people are like dandelions, able to thrive in almost any environment, while others are like orchids, who may struggle in harsh conditions but bloom spectacularly when the environment is supportive and nurturing. This distinction offers a biological validation for our personality differences, proving that emotional sensitivity is not a choice or a flaw, but a hardwired setting on the biological dashboard. I am curious to know if you consider yourself to be a highly sensitive person, perhaps feeling like the orchid that picks up on the subtle moods of a room instantly, or if you tend to be more like the dandelion, steady and generally unaffected by the emotional climate around you. Understanding this genetic variation encourages a deeper compassion for how we each navigate the world. It suggests that the intensity with which we feel joy, sorrow, or stress is partly dictated by the length of a microscopic protein chain. It reframes the human experience not as a standard operating procedure, but as a spectrum of chemical responsiveness where every point on the scale serves a purpose. The person who feels deeply is the sentinel, while the person who remains stoic is the anchor, and both are necessary for the stability of the whole. This genetic diversity ensures that within any group there is a balance of vigilance and resilience. However, the human drive to alleviate suffering meant that we were not content to simply observe these genetic dispositions. We wanted to intervene. This desire kicked off one of the most fascinating chapters in medical history: the quest to chemically modulate our moods by hacking the serotonin system. Interestingly, the story begins not with a grand design, but with a happy accident in a tuberculosis ward in the early 1950s. Doctors noticed that patients treated with a new drug called iproniazid, intended to fight lung infections, were experiencing a strange side effect. They weren't just breathing better, they were unusually energetic, sociable, and optimistic, despite being confined to a hospital with a serious illness. It was soon discovered that this drug inhibited an enzyme that breaks down neurotransmitters, effectively causing a flood of serotonin and norepinephrine in the brain. This accidental discovery ushered in the first generation of antidepressants, known as MAOIs, and shortly after, the tricyclics. While effective, these early compounds were often referred to by chemists as dirty drugs. In pharmacology, a dirty drug is one that doesn't just hit its intended target, but splashes onto many others, like using a sledgehammer to hang a picture frame. They boosted serotonin, yes, but they also interfered with histamine and other systems, leading to a host of sedative and physical side effects that made them difficult for many people to tolerate. The holy grail of neuroscience became the search for a magic bullet, a molecule that was precise, elegant, and selective. This search culminated in the 70s and 80s with the development of the SSRI, or selective serotonin reuptake inhibitor. To understand why this was such a revolution and to truly grasp how humanity managed to tweak the serotonin system, we have to look closely at the microscopic gap between our neurons, known as the synapse. Communication in the brain happens when one nerve cell releases a chemical message to its neighbor. Serotonin is released into the gap, it floats across, binds to the neighbor's receptors, and delivers its signal of stability or contentment. But nature is efficient and it hates waste. Once the message is delivered, the first cell immediately activates little pumps or transporters that act like vacuum cleaners. They suck the serotonin back up into the cell to be recycled and reused later. This process is called reuptake. Under normal circumstances, this cleanup happens very quickly, meaning the serotonin only stays in the gap for a split second. The genius of the SSRI was in its ability to selectively jam those vacuum cleaners. By blocking the reuptake pump, the medication prevents the serotonin from being recycled so quickly. It forces the molecule to stay in the synaptic gap for a longer duration, banging against the receptors over and over again. It effectively amplifies the volume of the serotonin signal without the brain actually producing more serotonin. It is a way of making the existing supply work overtime. When fluoxetine, the first major drug of this class, arrived on the market in the late 1980s, it fundamentally changed the cultural conversation around mental health. It shifted the perception of mood from a matter of pure willpower to a matter of biology. However, the story proved to be more complex than simply filling up a tank that was running low. Scientists eventually realized that while these drugs boost serotonin levels almost immediately, the mood-lifting effects often take weeks to manifest. This delay revealed that serotonin was doing something more profound than just cheering up the neurons. It was likely triggering a slow process of neurogenesis, instructing the brain to grow new connections and repair itself over time. This realization fundamentally shifts how we understand the very nature of mood and the brain, effectively dismantling the popular yet overly simplistic myth of a chemical imbalance. For decades, the prevailing narrative was incredibly straightforward, almost mechanical in its logic. We were told that the brain was akin to a car engine, and depression was simply the result of running low on a specific fluid, much like running out of oil or gasoline. If the tank was empty, the engine sputtered. If you filled the tank, the machine ran smoothly again. However, modern neuroscience has revealed that this picture is far too rudimentary to capture the elegant complexity of human consciousness. Extensive reviews of decades of data have shown that there is actually no consistent universal link between low serotonin levels alone and the onset of clinical depression. In fact, when researchers have artificially lowered serotonin levels in healthy volunteers to see what would happen, those volunteers did not automatically plunge into despair. This pivotal finding suggests that while serotonin is undoubtedly a critical player on the field, it is not the entire game. Instead of viewing the brain as a simple beaker of mixed fluids where the ratios must be exact, scientists now view it as a complex dynamic circuit board, or perhaps a vast adaptable ecosystem.
The issue isn't necessarily the sheer volume of the chemical messenger, but rather how the neurons are connecting, firing, and interpreting those messages. This brings us back to the vital concept of neuroplasticity and the neurotrophic hypothesis. The brain is constantly remodeling itself, pruning old, unused connections and forging new, vital pathways, a process that relies heavily on a protein called brain-derived neurotrophic factor, or BDNF. Serotonin appears to be a key regulator of this growth factor. When serotonin signaling is active and efficient, it encourages the production of BDNF, which in turn supports the survival and growth of neurons in critical areas like the hippocampus, the region of the brain essential for memory, learning, and emotion regulation. It is deeply fascinating to note that in individuals struggling with chronic stress or low mood, the hippocampus can actually physically shrink, losing volume as neural connections wither away. The recovery process, often facilitated by treatments that target the serotonin system, is therefore less about an instant chemical high and more about creating the biological conditions necessary for physical regrowth. It is the restoration of neural flexibility. The brain needs to relearn how to be resilient, and that biological learning process requires time, much like healing a bone or training a muscle. The serotonin is merely the signal that tells the cellular construction crew to get to work, rather than the building material itself. This nuanced view also helps explain why the biological reality of mental health is so individualized. Two people might have similar symptoms, but vastly different underlying circuit patterns, shaped by their unique genetics, environments, and life histories. The chemical environment is just the backdrop for a much more intricate performance involving electrical signals, structural changes, and a host of other molecules like glutamate, dopamine, and cortisol. We are finally moving toward an era where we see mental health not as a static state of chemical inventory, but as a measure of the brain's ability to adapt, change, and rewire itself in response to the world. It is a story of biological resilience and adaptation, rather than just a simple deficit and a refill. This capacity for change is perhaps nowhere more evident than in the strange and potent relationship between serotonin and the class of compounds we call psychedelics. To understand this interaction, we must look first at the geometry of the molecules themselves. It is a case of molecular mimicry. Psilocybin, the active compound in magic mushrooms, and LSD, a semi-synthetic substance, possess a chemical structure that is strikingly similar to serotonin. They both share the distinct indole ring, a double-ring structure containing nitrogen that acts as a skeleton key for the brain's signaling system. Because of this physical resemblance, these compounds can slip past the brain's chemical defenses and occupy the same receptors that serotonin usually patrols. However, they do not simply replace serotonin, they interact with the system in a fundamentally different way. While serotonin binds to a wide variety of receptor subtypes to regulate everything from sleep to nausea, psychedelics show a profound and specific affinity for the 5-HT2A receptor. This receptor is densely populated in the cortex, particularly on the deep layer pyramidal neurons, which are crucial for high-level cognition, perception, and the integration of sensory information. When a molecule like psilocybin binds to the 5-HT2A receptor, it does not merely activate it, it triggers a distinct signaling cascade that creates a state of hyperexcitation. If serotonin is a gentle conductor keeping the orchestra in time, a psychedelic compound is a new sheet of music that encourages every instrument to improvise at once. This leads to a phenomenon that neuroscientists describe using the entropic brain theory. In our normal, waking state, the adult brain is highly organized, rigid, and constrained. It operates on efficiency,
filtering out vast amounts of sensory data to present us with a stable, predictable version of reality. This stability is low entropy. When the 5-HT2A receptors are flooded by these serotonin mimics, the brain enters a state of hyperentropy. The usual filters dissolve, the rigidity relaxes. The brain becomes less like a structured hierarchy and more like a chaotic, fluid network. This increase in disorder allows for a dramatic rise in the number of possible neural states. Regions of the brain that have effectively been segregated since childhood, such as the visual cortex and the auditory cortex, suddenly begin to communicate freely. This crosstalk is the neurological basis for synesthesia, where sounds might be experienced as colors or emotions might be felt as physical sensations. At the center of this transformation is the dissolution of the default mode network. The default mode network is a collection of brain regions that are most active when we are not focused on the outside world, when we are daydreaming, worrying, remembering the past, or planning the future. It is the neurological anchor of the ego, the sense of a distinct self that is separate from the environment. Under the influence of 5-HT2A activation, the blood flow and electrical integrity of the default mode network plummet.
As this network disintegrates, the boundary between the self and the world fades away. The brain stops generating the narrative of I, leading to the profound sensation of oneness or oceanic boundlessness often reported in these states. It is a temporary rewiring that suggests our everyday perception of reality is a carefully constructed hallucination held in place by serotonin, and that by tweaking the chemistry of just one receptor, the entire theater of consciousness can be rebuilt. While classical psychedelics like LSD alter the visual and conceptual structure of reality, the compound known as MDMA targets the emotional architecture of our social existence, providing perhaps the most dramatic demonstration of serotonin's absolute power over how we connect with others. Under normal conditions, your brain operates like a frugal accountant, carefully releasing serotonin in measured doses and then quickly sweeping it back up using transporter proteins to be recycled. This system ensures that your mood remains stable and responsive to the environment. MDMA, however, completely subverts this delicate economy. It enters the brain and binds to those serotonin transporters, but instead of simply blocking them, it reverses their direction. It acts like a key that unlocks the warehouse doors and jams them open. The transporters, which are supposed to be vacuuming up excess chemical signals, begin pumping the brain's entire reserve supply of serotonin out into the synapses in a massive, uncontrolled flood. This results in a sudden, overwhelming saturation of the 5-HT receptors, triggering a cascade of neural activity that the brain interprets as absolute, unshakable well-being. The subjective experience of this serotonin deluge is often described as oneness or entactogenic, meaning touching within. As the serotonin floods the pathways connecting the amygdala, the brain's fear center, to the prefrontal cortex, the biological machinery of social anxiety shuts down. The fear of judgment, the instinct to protect one's ego, and the subtle defensiveness we carry in social interactions simply evaporate. Users report feeling a profound, limitless empathy for strangers, a sensation that the barriers between self and other have dissolved. It reveals that the feeling of separation we experience in daily life is, in part, chemically maintained, and that by tweaking the serotonin dial, the brain can be convinced that everyone is family. However, this chemical loan comes with a steep interest rate. Because the drug forces the neurons to dump their stored supply of serotonin all at once, the brain is left temporarily bankrupt. Once the drug wears off, the reservoirs are empty, and the production of new serotonin is a slow biological process that can take days or weeks. This leads to a phenomenon famously known as the Tuesday blues, a period of low mood, irritability, and social withdrawal that typically hits a few days after the weekend use.
During this depletion phase, the contrast becomes stark. The same world that felt full of love and connection just 48 hours prior now seems cold, gray, and hostile. Nothing about the external environment has changed. The only difference is the absence of the molecule that colors our social perception. This cycle of flood and drought serves as a profound lesson in neurochemistry. It demonstrates that our ability to trust, to bond, and to feel a part of the human whole is not just a spiritual choice, but a biological function dependent on the availability of a specific neurotransmitter. It suggests that serotonin acts as the invisible glue of society, a chemical signal that tells us we are safe among our kind. While this social glue binds us to one another, the way we acquire it for our own brains is surprisingly complex, leading to one of the enduring myths in the world of nutrition. It is a common belief that eating foods rich in serotonin, such as bananas, walnuts, pineapples, or tomatoes, will directly increase the levels of this chemical within your mind and subsequently lift your mood. The biological reality, however, is far more restrictive and fascinating. The serotonin found in these foods is chemically identical to the serotonin in your head, but it is effectively barred from entry by the blood-brain barrier. This protective shield, a network of tightly packed cells lining the capillaries of the brain, acts as a vigilant gatekeeper designed to prevent foreign substances, toxins, and certain neurotransmitters circulating in the bloodstream from disrupting the delicate chemical balance of the central nervous system. When you consume a serotonin-rich banana, the chemical is metabolized in the gut or remains in the general circulation, where it might influence digestion or blood vessel constriction, but it never crosses the threshold into your brain to alter your emotional state. Instead of importing the finished product, the brain must manufacture its own serotonin from scratch, using raw materials imported from the diet. The critical ingredient for this synthesis is tryptophan, an essential amino acid that the body cannot produce on its own. This creates a logistical challenge because tryptophan is essentially a VIP guest that has to fight through a crowd to get into the club. In the bloodstream, tryptophan must compete with other amino acids, specifically the large neutral amino acids like leucine, isoleucine, and valine, to gain passage across the blood-brain barrier.
They all utilize the exact same transport mechanism, a specific carrier protein that acts like a revolving door with limited capacity. Since tryptophan is relatively scarce in most protein-rich foods compared to these other dominant amino acids, it often gets crowded out, left waiting in the bloodstream while the others are ushered into the brain. This leads to a counterintuitive fact regarding diet. Eating a high-protein meal, which contains plenty of tryptophan, can actually result in lower brain tryptophan levels because the massive influx of competing amino acids blocks the transporter. This competition mechanism explains why we often crave carbohydrates rather than protein when we are feeling low or stressed. The consumption of carbohydrates triggers the release of insulin, a hormone that instructs the body's cells, particularly muscle tissue, to absorb amino acids from the blood. Crucially, insulin clears out the competitive large neutral amino acids, sweeping them into the muscles, but it leaves tryptophan largely untouched in the bloodstream. With the competition suddenly removed, the path to the brain is clear. The ratio of tryptophan to other amino acids shifts dramatically in its favor, allowing it to occupy the transporters and cross the blood-brain barrier in significant numbers. Once inside the sanctuary of the brain, it is quickly converted into 5-hydroxytryptophan and finally into serotonin. This biological loop validates the existence of comfort food, providing a scientific basis for why a bowl of pasta, a slice of toast, or warm oatmeal can feel chemically soothing in a way that a steak does not.
It is an unconscious form of self-medication where we manipulate our own insulin levels to engineer a clear path for serotonin production. Speaking of those quiet moments of nourishment, I am curious to know what simple comfort food or evening routine helps you unwind the most when the day is done. Understanding this pathway reveals that the link between what we eat and how we feel is not about simply consuming the chemical we desire, but about understanding the intricate traffic laws of our internal biology. It suggests that the brain's serenity is often purchased with the currency of carbohydrates, acting as the strategic facilitator that allows the building blocks of happiness to reach their destination. This strategic intake of food highlights the brain's dependency on the body's metabolic state, proving that our mood is inextricably tied to the simple, rhythmic processing of nutrients. Just as the gut works in cycles, the brain responds to the physical cycles of the body. One of the most potent triggers for serotonin release is simple, repetitive motor activity. Scientists have observed that when we engage in rhythmic motions like walking, cycling, or even chewing, the firing rate of serotonin neurons in the brainstem increases significantly. This suggests that the brain interprets steady physical rhythm as a signal of safety and active engagement with the world, prompting it to upregulate the chemicals responsible for mood stability. It is not necessarily the high intensity of a workout that matters most for this specific chemical release, but the consistent, repetitive pattern of the movement itself, which acts as a metronome for the mind. This mechanism is a key component of the phenomenon often called the runner's high. While popular culture has long attributed this state of euphoria solely to endorphins, modern research reveals a more complex symphony involving endocannabinoids and serotonin working in tandem. The sustained rhythmic impact of running stimulates a profound release of serotonin that persists long after the physical exertion has stopped, contributing to the afterglow effect, where stress feels manageable and the mind feels clear. This is the body's way of rewarding perseverance, using chemistry to encourage the continued movement necessary for survival. However, the setting in which we move is just as influential as the movement itself. There is a specific biological response to being in nature, a concept codified in Japan as Shinrin-yoku, or forest bathing. When we breathe in the air of a dense forest, we are inhaling phytocides, which are airborne antimicrobial organic compounds released by trees to protect themselves from insects and rot. When humans inhale these compounds, they trigger a cascade of physiological changes, most notably a significant reduction in cortisol, the primary stress hormone. Since cortisol and serotonin often work on a seesaw, where high stress dampens serotonin function, lowering cortisol naturally allows the serotonin system to work more efficiently. Have you ever noticed how different the air smells in a dense forest compared to a city park? And does that scent instantly change your mood? This natural regulation extends beyond where we are to how we interact with others. Physical touch is another ancient modulator of this neurotransmitter. Studies on massage therapy have shown that a sustained session can increase serotonin levels by nearly 30%, while simultaneously decreasing cortisol by a similar amount. This reaction is likely mediated by the vagus nerve. When pressure receptors under the skin, known as Pacinian corpuscles, are stimulated, they send signals to the vagus nerve to slow the heart rate and lower blood pressure, shifting the nervous system from a state of fight or flight to rest and digest.
In this relaxed state, the brain is primed to replenish its serotonin stores. This mirrors the grooming behaviors seen in primates, where social bonding through touch is essential for maintaining group harmony and individual calm, proving that our neurochemistry is designed to be responsive to the gentle pressures of the outside world. However, this delicate responsiveness relies entirely on precision, because even a molecule associated with happiness, sleep, and calm has a strict upper limit. We might intuitively assume that if a small amount of serotonin makes us feel stable, a massive amount would make us feel euphoric. But the biology of the brain tells a starkly different and more cautionary story. There is a specific condition known as serotonin syndrome, a serious medical state that arises not from a deficit, but from an overwhelming surplus. It serves as a potent reminder that our internal ecosystems are governed by the law of the golden mean, where true health is found only in the middle ground. When the serotonin reservoirs overflow, usually triggered by the accidental combination of medications or supplements that prevent its reuptake or breakdown, the synapses become flooded with more chemical messengers than they were designed to handle. Instead of a gentle, rhythmic signal promoting well-being, the nervous system receives a deafening roar of chemical information. The result is a cascade of chaos known as autonomic instability. The body's internal thermostat essentially breaks, causing body temperature to rise rapidly, while the heart beats faster and muscles become rigid or twitch involuntarily, a phenomenon doctors call clonus. This is the nervous system operating at hyperspeed, unable to find the brakes, racing forward without a driver. It is a biological paradox where the very chemical that regulates our sleep cycles and digestion turns against the body's basic housekeeping functions simply because the volume has been turned up too high. Scientists have observed that this toxicity specifically overstimulates the 5-HT2A and 5-HT1A receptors, essentially jamming the locks by forcing in too many keys at once. The existence of this syndrome illustrates a profound truth about human physiology. The ultimate goal of the brain is never maximization, but homeostasis. It strives for a steady, unwavering state of equilibrium. Just as water is essential for life, but a flood is destructive, serotonin is vital for mood, but toxic in excess. The body is constantly working to metabolize and clear out old serotonin to prevent this dangerous accumulation, engaging in a perpetual cleaning cycle to ensure that the chemical remains a messenger of balance rather than an agent of disorder. This built-in biological safety valve highlights the incredible complexity of the pharmacological tightrope we walk every moment, often without ever realizing the silent, constant adjustments keeping us safe from our own chemistry. Ultimately, the story of serotonin is not just about a single neurotransmitter, but about the profound interconnectedness of life itself. We began this exploration looking for a simple chemical of happiness, yet we discovered an ancient molecular architect that constructs the very foundations of our reality. It is a molecule that bridges the vast divide between the physical and the mental, synthesized from the raw nutrients in our food to power the complex ethereal machinery of our dreams. From the millions of neurons lining the digestive tract, ensuring our inner biological rhythms keep perfect time, to the microscopic gap of the synapse where a single spark can determine whether we feel frustration or calm, serotonin is the silent conductor of the human symphony. It is humbling to realize that our ability to navigate the world, to tolerate the friction of daily life, and to drift peacefully into sleep relies on this delicate dance of atoms, carbon, hydrogen, nitrogen, and oxygen arranged in just the right way.
This ancient messenger, shared with the walnuts we eat and the nettles that grow in the wild, serves as a lasting reminder that biology is incredibly efficient, reusing its most effective tools across eons of evolution to sustain life. It filters the chaotic noise of the external environment, dimming the harsh lights of sensory input so that we can focus, learn, and eventually transition into the restorative darkness of the night. As we conclude this exploration of the body's most fascinating chemical balancing act, I hope you have found a sense of wonder in the quiet complexity of your own biology. If you have found a moment of calm in understanding the chemistry of your own mind, I invite you to subscribe to this channel, ensuring we can continue these relaxing investigations into the natural world together. For now, we are left with the comforting thought that deep within us there is a system entirely dedicated to maintaining equilibrium, a tireless mechanism working in the background to ensure that after every period of activity there is rest, and after every challenge there is the potential for peace. It is the molecule of balance, holding the weight of our consciousness, allowing us to close our eyes and trust that we will wake up ready for a new day.